There is a protein which can form pores inside our body cells. Imagine if that goes wrong. Hello, I'm Anne-Rosa and I am fascinated by this protein, Perforin, which helps my favorite cells, cytotoxic T cells, a type of white blood cells, to eliminate virally infected body cells. The underactivity of this protein can cause a family of genetic diseases called familial hemophagocytic lymphohistiocytosis, associated with limited immune response to infections. On the other hand, the overactivity of this protein can be linked to systemic lupus, with high levels of inflammatory cytokines which could engage the immune system to the point of exhaustion. I found that there is evidence to suggest that studying these two family of diseases could increase our understanding of COVID-19. That is because in some patients with poor outcomes post-infection, there is a very limited immune response with low numbers of cytotoxic T cells and low concentrations of perforin. However, on the other hand, there is a group of patients that experience poor outcomes with an exaggerated immune response and too high a perforin concentration as well. A common denominator between these two families is perforin. Although a very big study analyzing the whole genome in relation to perforin did not find direct genetic links between worse outcomes and the genes that code for perforin, researchers have concluded that perforin still does contribute, at the very least indirectly. So seeing that this protein seems to have such a footprint on COVID-19, it was not a surprise to discover that it could be used to boost the cellular immune response itself through the use of a type of vaccine called a DNA vaccine. This comes at a very useful time because there is still global COVID-19 inequity. In the past year, there has been a mere increase of only 20% of people receiving a COVID-19 vaccine in low-income countries, which is really disheartening. And to top it, 50% less people have received the vaccines in LICs anyway. This vaccine itself is not only easier to manufacture than contemporary ones, but also easier to transport because of its high stability. This could help us get the vaccine to areas with low access, especially in LICs. However, unfortunately, 95% of the DNA content in this vaccine does not reach its intended target cells. I have investigated evidence to suggest that perforin could be used to counter the effect of this by ensuring that the DNA that does enter the cell successfully can have maximum impact. After 3 years of starting this project, luckily now there are vaccines in development that are trying to implement this technology, including using perforin as an adjuvant, already with signs of success. This is important because not only do we want to bridge the vaccine equity gap, we also want to be able to extrapolate our newfound understanding to help us tackle similar viral diseases more successfully than COVID-19 in the future.